working in this beautiful institute in Bordeaux, France. And as I said, our work has been focused on electrospray mass spectrometry to study nucleic acid complexes. So this comes from a quite old review. So the principle is that we are mixing the DNA target, can be quadruplex, can be something else, and ligands. And the most important thing to remember with mass spectrometry, maybe the one big take-home message, is that when you're reading a mass spectrum, you are really reading the stoichiometries of the complexes that you form. So I will take a minute or so to explain you also how to read the mass spectra, because it's overall quite easy. Because what we do is we start from oligonucleotides that we purchase. So we know the mass of the oligonucleotides. And on the mass spectrometry scale, you always have a mass divided by charge on the scale. So it means if you have a given mass, here D is for a duplex, you have the mass of the duplex, and you have several peaks for D with different numbers of charges. So divide by 4, by 5, or by 6 charges. And the charges is just a number of phosphate groups that have not been completely neutralized by protons coming from the ammonium ions. So here we have 3 peaks. It's the same molecule. It's the duplex alone. And then for each of the charge states, we have a distribution of complexes with 1 and 2 ligands bound. So again, we know the mass of the ligand that we add to the solution. So we can just calculate the theoretical sum of the masses for all the complexes that are possible. And then you divide by the different numbers of charges that would be possible to assign each of these peaks. So for us, when we read a mass spectrum, it's not a fitting to determine the stoichiometry, it's just reading the masses on this x-axis. And then, of course, with the intensities, we can tell something about the quantities. So the first thing with stoichiometries is the strength stoichiometry, because we are usually told in basic introductions on G-quadruplexes that if sequences have 1 G tract, you need 4 of them to form a tetramolecular G-quadruplex. If your sequence has 2 G tracts, you can form bimolecular structures. And if it has 4 G tracts, it can fall intramolecularly. But with our mass spectrometry investigations, we have also found some other behaviors that contradict this principle. For example, this is the example of a DNA G-quadruplex forming sequence from the telomeric sequence with 4 G tracts. In the DNA strands, so we are working, actually in the past work, we have been working a lot with ammonium acetate, where G-quadruplexes can fold and also is perfectly compatible with mass spectrometry. So DNA in these conditions fold intramolecularly. But when we studied RNA, the RNA version of the telomeric sequence, also 4 G tracts, the surprise was to find bimolecular assemblies of G-quadruplex. So 2 of such sequences assemble and make a dimer. So this was on the occasion of a collaboration with Steve Needle's lab. I see some message appearing. Okay, whatever. I see a message appearing on my screen from the Zoom. Okay, another example of unexpected result. No, it doesn't change. Okay, doesn't change slide. Another example of unexpected result is when we studied TG4T, TG5T, and we extended the number of bases in this TGNT tract. Initially, we wanted to have some molecular ruler, just a long quadruplex where we could tune the length, but it turned out not to work in that way at all. So when we extend beyond a certain length, actually, we don't have any G-quadruplex forming, so no tetramolecular complex forming. And starting with some length, we can have some dimers, some trimers forming. And then when we do the annealing in 10 millimolar ammonium acetate, the surprise was that we have only intramolecular G-quadruplex forming once we have TG15T. And we also detected the binding of 2 ammonium ions in this species, and really starting from 15 Gs, which we interpreted with a cartoon at the time, imagining that we have 12 Gs involved into a 3 tetrad G-quadruplex, And then 3 of the Gs are constituting the loops that join these G threads. And this, based on this cartoon, actually Antoine Fan later studied this structure, this sequence by NMR, and found exactly at high resolution that the topology we kind of predicted thanks to mass spectrometry was confirmed in the high resolution structure. And we've also seen a difference of the annealing conditions. If you use low ionic strength or high ionic strength, you are going to form different stoichiometries if you anneal those sequences. For example, at high ionic strength, you are not forming an intramolecular G-quadruplex, but you are again forming a bimolecular species of this TG15D. Here's just a cartoon of something that could be compatible with the observations, but there are several topologies that could actually fit. So it's an example where actually mass spectrometry revealed let's say an unpredicted result, but which turned out to be important for say expanding our knowledge of biophysics and how G-quadruplexes can form, just based on measuring the number of strands in the fold. A second topic is going back to the telomeric G-quadruplexes. Actually, that's how we started to get into this field. Well, it's a bit more than 20 years ago now. That we wanted to have a technique to screen ligand binding to telomeric G-quadruplexes, and at the time there were these 2 structures, the NMR structure in sodium, the X-ray crystal structure in potassium, which are hugely different. Then later on, it turned out that several other structures could be solved in potassium solution based on the beginning and terminal bases of the sequence you chose to resolve the NMR structure. So we know that the exact sequence is important. We also know that depending on the number of G tetrads, you need cations to fit in between the G quartets in order for the structure to form. And also, I told you in the early work on mass spectrometry, we have been using ammonium acetate. That's what everybody was using for proteins in a native state. And G-quadruplexes fall in ammonium. But for example, the telomeric G-quadruplexes are so sensitive to the cation that we knew from CD it was not exactly the same fold for the telomeric G-quadruplexes in ammonium and potassium. So one of our goals was to be able to do also mass spectrometry in potassium conditions, specifically for telomeric G-quadruplexes. So when I started in Bordeaux, actually Adrien Marchand came up with an idea to solve this problem, being able to record mass spectra in potassium. Actually, we cannot put 100 millimolar potassium like you would do in other biophysical techniques in mass spectrometry. The spectra would be just awful. But we found out that we could ensure the ionic strength using trimethylammonium acetate instead of ammonium acetate. So trimethylammonium is too big to go in between G quartets, and it will just serve to ensure the ionic strength, but it will not bind to G quadruplex. But then you can add small amounts of potassium, up to 1 millimolar. You still have a nice mass spectra. And this is enough for the G quadruplexes to fold to an extent that is compatible with what we wanted to do. So again, how do we read those mass spectra? Here we mix a strand that is a derivative of the telomeric sequence, small amount of potassium. Again, we have 3 sets of peaks, and this reference here is just a T6 short oligonucleotide we use for the reference of the peak height. So we are not going to use it at this moment. But each of the peak, if you zoom on this on each of the charge states, we actually have 3 peaks hiding there. And thanks to the mass resolution of a mass spectrometer, you can distinguish the binding of 0, or 1, or 2 potassium ions to your sequence. So it is just so high resolution, something you could never see in a gel, for example. So we can have these stoichiometries, which we can relate then to the number of G quartets involved in these structures. Then another thing was to do this quantitatively, and especially make sure, sorry, the scale does not appear on this slide. So it's a titration experiment, several samples that were injected between 0 and 1 millimolar of KCl. This is 1 millimolar, this is 200 micromolar you saw before. We increased the amount of potassium, but we also wanted to make sure what to do with these signals, because 3 potassium ions doesn't really make sense in a sequence that has only tracks of 3 guanines. So we also run titrations in the same conditions with a control sequence that is unable to form the G-quadruplexes. There are as many Gs, but they are randomized in the sequence. And then we can deduce the amount of signal that is due just to non-specific binding to the sequence. It means non-G-quadruplex specific binding. So it means that this amount of signal we can mathematically subtract from here to reconstruct the amount of species with 0 potassium, so unfolded, 1 potassium, it means it's 2-quartet ensemble, and 2 potassium, which is a 3-quartet ensemble. So we can then have these data points is amount of DNA of each form as a function of KCl concentration. And you can use fitting algorithms to use the KD value, but for each of the stoichiometries that you can distinguish with mass spectrometry. And in this 2016 paper, you can see how it is very different depending on the exact sequence that you choose. So again, changing the first or the terminal bases. These 3 sequences correspond to 3 sequences for which high resolution NMR structure were solved. These actually, it's the same hybrid 1 final structure, but you can see that in terms of binding affinity and binding cooperativity for potassium, it's very different. This sequence 24TTG is binding to potassium ions very cooperatively with high affinity. This is the one you saw before, and there is at intermediate potassium concentration a significant amount of 2-quartet species, just 1 potassium. And this sequence 22GT is the one that is known to fall into a 2-quartet quadruplex and then a tetrad on top of it. And then we can see a lower affinity, a second binding site for the second potassium, presumably between the triplet and the quartet. So all this is detailed in this paper. You can see the variance. You can see also the effect of fluorophores that you typically use in fret melting experiments. Actually, the fluorophores have a big influence on the thermodynamics of potassium binding and folding. Now, another thing you can do by knowing the amounts of each species you have with mass spectrometry is when you do another biophysical experiments, for example, CD spectroscopy in exactly the same range of concentrations, you can use the amounts of species of each state that you know from mass spectrometry and you have quantified from mass spectrometry to deconvolute the CD signals for the 3 states that we can distinguish with mass spectrometry. So of course, the first state, the unfolded state, is easy to extract because it's the experiment you would do without KCl. The final state is close to the spectrum you will get with the maximum amount of KCl. But what would be difficult just from the CD spectrometry, we kind of guess here, it's not just 2 states, but it's hard to extract the reconstructed CD spectrum of the intermediate species. And this is what we can do by knowing the amounts at each point in the titration. And when we reconstructed this CD signal, we found the signature of an antiparallel type of stacking, so heterostacking. So this is how we can draw this cartoon for this species form at intermediate KCl concentration. We know it has 2 quartets because it has 1 potassium, but we also know that it's a heterostacking and presumably an antiparallel quadruplex structure. And what we can do also to go a bit further with mass spectrometry experiments, I showed you thermodynamics, KD determination. We can also do kinetic experiments, so time-dependent experiments. And here we did this on the sequence, which we call 24TTG. So it starts with T's, it ends with A's. It's the one that forms very cooperatively the 2 potassium quadruplex, the hybrid 1 with 3 quartets. But that only forms actually quite slowly. If you mix at room temperature, when you start the mixing, adding the potassium to the G-quadruplex sequence, it actually takes nearly an hour to fold correctly into the final structure. And we have, again, a species populated at intermediate times, which is a 1 potassium species. It means it's a 2 quartet species. And again, when we extracted the CD signal, we found out it's antiparallel. So then this led us to propose and discuss this folding mechanism for G-quadruplexes It's not a stepwise formation incorporation of one potassium and then a second potassium. Actually, it's a branched pathway of folding. An unfolded strand, the strand that we start with, it can grab quite readily because these form fast. It can grab right readily a first potassium ion, but we have an ensemble of structures with misaligned strands. So it's probably an ensemble of different structures there that can form fast, but for many of the sequence is not the thermodynamically most stable structure, which would be the hybrid 1 or the hybrid 2. And these form actually slower than the 2-quartet antiparallel structures. So more recently, we have been studying a number of sequences with these conditions in TMA and 1 millimolar KCl, because we wanted to document a list of sequences in those conditions where you could know which structure you have in the mass spec conditions. So we took over 30 sequences for which NMR structures had been published in 100 millimolar KCl or something like. And we performed CD spectroscopy in green, it's always the NMR conditions, so the 100 millimolar KCl. In orange is in our mass spec conditions, so 1 millimolar KCl and TMA. And here is the unfolded one. We also documented the NMR spectra in the mass spec conditions that you can verify it's the same structure as in the high KCl concentrations. And we have documented a database, so it's both in this paper and on Eric Largy's personal website that you can, here you can browse a bit more smoothly. And you can see which sequence you might want to include in a screening assay, for example, and have here the validation, what extent the structure in mass spec conditions match those you would know from NMR in high KCl conditions. And most of the times when we don't have a really good match of the CD spectra, it's more a matter of intensity of amount formed. For example, with this sequence referred to by its PDB code, we have the same NMR peaks as for the fully folded structure. It's just that the extent of folding is not 100%. So we have small amounts of unfolded without potassium. And here a bit of species with 1 potassium as well. So you can use this database if you want to conceive screening assays in those mass spec compatible conditions. Now, a few other examples then pick from various papers on another type of cation binding experiment that you can do with mass spectrometry is actually cation exchange. And this was from a collaboration with what we had with Antoine Fan on the ZG4 sequence. So actually the ZG4, it has exactly the same fold when in potassium or ammonium conditions. So then it was a good system to test with the same structure, the kinetics of exchange between ammonium and potassium, for example. So this paper is about cation residence time into the ZG4, which we found it's very long residence time, especially for the central cation. So we can do that because ammonium and potassium have different masses. Here's the starting point with 3 potassium ions bound. Ammonium is a bit lighter, and actually in each of the peak you can assign how many how many ammoniums and potassiums you have. And what we found is that the exchange of the first 2 potassiums for ammonium is very fast. In 2 minutes it's done, and we believe it's the top and bottom ones. But actually the central cation takes several hours to be exchanged for ammonium. So this one of the major findings of this paper. And then while we were submitting that, one reviewer asked, but what does it tell us about the exchange between potassium ions themselves? And we scratch our head a bit, because then we have potassium for potassium. But actually, it turns out that you can purchase a different isotope of potassium. It's a bit more expensive than the natural KCl that you would buy from the shelf, which is the mainly 39 potassium. But we bought 41 potassium, did the annealing, have the quadruplex form with 41 with potassium 41. and then could monitor the K plus K plus exchange as a function of time. And then with isotope exchange of the same cation, we could monitor potassium exchange. So it was a nice application of mass spectrometry. And I really like to thank Antoine Pham, because actually he came up with this idea of buying the heavy potassium. And by the way, and also a little advertisement for another technique. Now it's not measuring isotope exchange of the inner cations, but it's measuring the isotope exchange of the hydrogens for deuterium. Actually, hydrogen deuterium exchange, MS, is pretty widespread for studying protein biophysics, but it had never been used before for studying nucleic acid biophysics. And when he started in the lab, Eric Largy, that's what he started doing. Of course, we knew from NMR that hydrogen deuterium exchange can be slow. It's even a marker of G-quadruplex formation, the fact that you have the slow exchange. But how could we translate that to mass spectrometry? And here the beauty is that when you have several states of folding that you can distinguish by mass, for example, by the number. Again, it's a sequence in a potassium concentration when you have coexistence between these three forms with different number of quartets. You can measure the rate of HD exchange independently for each of these stoichiometric states of potassium binding. So here you have a slow exchange when it's fully folded into the three-quartet quadruplex, almost instantaneously exchange for the unfolded sequence, and something intermediate for these two-quartet species. Then you can see that as a function of, this is plotted as a function of time. So there are several papers still in preparation for the real-life application to biophysics now and to ligand binding to G-quadruplexes. Okay, so I'm now going to switch to the characterization of ligands binding to G-quadruplexes. So I showed you can detect number of strands, number of cations down, but now what about the ligand binding to each of these species? And a question we had very early on is whether we could rely on the relative intensities in the mass spectra to assume that the intensity ratio was proportional to the concentration ratio in solution. Basically, it's an assumption that has always been questioned in mass spectrometry, like can you ionize with the same efficiency different forms of a DNA, for example. And in this paper, we have shown how we can test for this hypothesis just by using this internal standard I was referring to earlier. So actually, we always inject this small sequence in our samples. We have this small oligo T6 to which most drugs don't bind. And then we do the titration experiments with always these T6 strand inside. So it's like a scale of reference. And we can monitor if all the intensities are accounted for when normalized to T6. If the sum of intensities is the same, it means the assumption of equal response factor is satisfied. And if it's not the same, we can actually use the T6 signal to correct for the relative response. And that is what, and that is a procedure described in this earlier paper. But, and what we found is that most of the time, actually the hypothesis of equal response is satisfied in the conditions we work in. So to understand that, just let's have a little intermezzo with the on the electrospray mechanism itself, because I didn't explain yet how the experiments were carried out and how it is that we produce ions desolvated from the solution. So the principle of electrospray is we inject a solution with a syringe or and in a thin capillary that is metallized, so we have conductance. And then when we apply a voltage or difference between the capillary that contains the solution and the counter electrode which is down there, we have charges accumulating at the tip of the meniscus. So actually the meniscus is deforming, and at some critical point, it's emitting very small droplets at the end of this, at the end of the capillary that contains the solution. The thing is that the droplets here are charged because it's expelling the excess charges just to maintain the current between the capillary and the contour electrode. So the first step of the electrospray process is this charged droplet emission. So the droplets in negative mode, they are enriched in negative ions, and this is what we want for DNA, because DNA is negatively charged in solution with the phosphates. Now this is the production. There are some regime in electrospray where you can produce a very thin beam of very narrow droplets. And that's what happens next, because here droplets still contain millions of water molecules. So what happens next is that droplets themselves, they evaporate because we have still a lot of gas. It's ambient temperature and ambient pressure. Droplets, they collide with the gas. And at some point, they evaporate to the point where they are themselves unstable and eject themselves little streams of secondary droplets. So we have these several steps of droplet fission, and actually it's uneven fission because it produces not breaking in two parts, but really producing a jet of smaller droplets. So these processes repeat themselves only a few times, and then you get to very, very tiny droplets containing only one ion. So one ion of your DNA. And then what can happen with your the final production process of your ions? These are basically the three mechanisms that have been proposed for the final production of desolvated ions from a charged droplets. The cartoon is for positive because most people study electrospray in positive mode, but it's actually the same for nucleic acids. I will show later. The first model is ion evaporation. It means that the ion can be expelled from the droplets, but actually this is not valid for large molecules like DNA. Maybe it's valid for acetate ion that can be expelled from the droplets, so small ions like sodium, acetate, etc. This may be valid, but not for a big nucleic acid. What happens for big molecules, proteins or nucleic acids, is basically these two extreme cases. It's called either a charged residue. So charged residue means that we have a droplet containing our biomolecule in the center. It's going to evaporate progressively until we have the bare biomolecule remaining at the end. So and then it grabs the remaining charges. So this is a charged residue, and the other mechanism is a chain ejection. This is more valid if you have an unfolded structure, especially for proteins, so a denatured protein, for example, or a denatured polymer. If it can go sit on the surface of the droplet, then it can also be expelled, and with charge rearrangement, it can grab a large number of charges and end up extended in the gas phase. And actually, this process is pretty efficient for charging molecules to a large extent. But for nucleic acids, basically what we want, if we want to do native mass spectrometry and study the structure as preserved as possible, basically we want every nucleic acid complex to end up in different droplets, and then each of the droplets, you want them to be produced with the charged residue mechanism. Then if you want to study gas phase structure, what you also want to avoid is extra compaction or extra extension after the charged residue. I may tackle that in the ion mobility section, but basically the first message is that you want to have this charged residue mechanism. If you have chain extrusion, there is high chance that it's going to unfold, and it may disrupt the native interactions and lose the specificity of your complexes, despite you would get higher signal. So you have to make a trade-off. And this is why it works actually that we have equal response factors most of the time with high ammonium acetate concentrations, is because if we use high ammonium acetate concentrations, we are coating the entire surface with acetate ions as charge carriers. It forces the nucleic acid to stay in the center, and it favors this charge residue mechanism. If you use too little amounts of ammonium acetate, you may then have this ionization with chain extrusion, and that's what we discuss in both of these papers. So now back to typical screening for ligand binding to a nucleic acid. Here it's not a G-quadruplex, it's an aptamer, but we studied that with Stefano as the most extreme case where we thought we would have huge differences in electrospray response because aptamers are supposed to wrap around their target. So the non-bound and bound structures of the aptamer are supposedly quite different, and then we expected differences in response. And then he did all this quantitative analysis of response factors for many aptamers, and basically what he found is that the correction for response factors that we typically have to bring is usually very, very small. It's actually the response factors are very similar. Here, the most extreme cases, it was a factor of 1.25, meaning that we would do on the KD value an error of a factor of 1.25, which is, as you think of it, is a very small error, because what you are interested in when you screen for ligands is huge difference in KD. So differences basically in log of the KD values. So here, this is a small correction if we correct for the response factor, and this is a very good match with the order of magnitude of binding constant that you would get from ITC. So from now on, we will use the approximation that response factor are equal between free and bound species of DNA. And if there is a little error, actually, you are looking for big differences. It's not a big deal. So now I'm going to show a few examples where by electrospray mass spectrometry studies of ligand binding, actually looking at little details in the mass spectra, we could find hints about the binding mode that we couldn't get with other techniques. So for example, this is the ligand copper TTPY. So when we bound it to a ternary quadruplex sequence, is the sequence that forms the 2-quartet species. Actually, what we found is not a 1-to-1 complex, but exclusively a 2-to-1 complex when the ligand bound. And also, when we zoom in on the peak here, we know that we are forming a 3-quartet quadruplex because it has 2 potassium ion bonds. The CD in the same conditions indicate that the final structure is also antiparallel. So we know it has 2 ligands, 2 cations, so 3 quartets, and that it's antiparallel. And again, we could propose a scheme, a cartoon of a possible ligand binding topology, just based on mass spectrometry data. Here's also another example where we can detect a ligand-induced quadruplex folding. And here it's with a sequence. Initially, we didn't really expect this one to make a quadruplex. And actually, when left alone, just in KCl, it doesn't make anything. The major species is with no potassium bound. But when you add FANDC3, again, a cooperative binding, there's not 1, but 2 FANDC3 molecules suffice to make this sequence. Well, here the cartoon, I don't really know the sequence, except that, again, it's antiparallel. That it folds in, it manages to grab a cation and arrange its guanines to form a 2-quartet G-quadruplex of some sort that is, again, antiparallel. Another example of details we can detect is cation grabbing with the ligands. This is an example with L2HU2-OTD molecule, so a microcyclic ligand. This is the published structure. And actually, you have an extra binding site possible between a quartet and the macrocyclic ligand, which is what we can detect in the peak of the complex. Free DNA has 2 potassium, 3 quartets, but when you grab the ligand, you have 1 more potassium ion binding. So we can propose this little cartoon. No change of structure compared to the starting quadruplex, but then an extra cation grab between the ligand and the quartet. And the final example is also well known. It's actually the opposite. When we bound the ligand, we saw the ejection of a potassium ion. And actually, the surprise came because it's some of the most widely used ligands, like FANDC3 or 360A or pyridostatin. You start with a telomeric G-quadruplex. It has a very stable hybrid 1 structure, but when you bind the ligand, the peak actually that is most intense has only 1 potassium, not 2. So it has 2 quartets, not 3. Again, CD shows its antiparallel. So at the time, we had this little figure in the paper showing different cartoons that could be compatible with the observations. 2 quartets, 1 cation, 1 ligand, antiparallel, well, heterostacking. And if you want to find out which of these 3 it is, you will have to listen to the presentation of Anirban tomorrow. Actually, you will find it's none of the cartoons that we initially proposed, and he will walk through the structure, because actually the hint with mass spectrometry was that this would be really interesting to solve at high resolution, and he actually cracked it with NMR in collaboration with Janis Plavic's group. All right, another type of experiment we can do is also a competition experiment. It's a bit like we did with the cations, different masses of cations you can detect coexisting on the quadruplex. And actually a very typical competition experiment among ligands that people do in biophysics is displacement experiments, for example, thiazole orange displacement assay. And a few years ago, we collaborated with Jean-Louis Mernier and Mauro Freccero. They had studied these molecules and actually had inconsistent results for the naphthalene diamide derivative binding to G-quadruplexes, inconsistent result between direct assays and the displacement assays. So it was not really clear. This molecule looked to bind, but actually at the same time not displace thiazole orange. So we decided to study that with mass spectrometry and to detect if there were any ternary complexes detected. So the first mass spectrum is just a G-quadruplex. So it's a mixed derivative with thiazole orange. We have the binding of thiazole orange 1 and 2. And then we mix with the naphthalene diamide, and now the spectra are becoming a bit more complicated, but thanks to the differences of mass, we can distinguish them. So the first peaks here is the same as in the spectrum above. Amount of G-quadruplex has diminished, of free G-quadruplex has diminished a bit, but we still have a G-quadruplex with thiazole orange. So actually, it's still bound. Then we have some peaks of G-quadruplex with the NDI alone. So it's what we have consumed, the free G-quadruplex that we have consumed, +1 and +2 NDI. But the most interesting was that we could have these ternary complexes here, the G-quadruplex with both a thiazole orange molecule and the NDI ligands presumably on the other side. So this is an example where with mass spectrometry, we could understand why there was an apparent discrepancy between the displacement assay and the direct binding assay. Actually, it can do both, bind the thiazole orange on one side and the NDI molecule on the other side, and that you can readily detect with mass spectrometry. Okay, and now a final layer of information that we can add with a mass spectrometry, a special experiment in mass spectrometry. Everything that I showed you up to now was done with room temperature experiments, but we can also control the temperature of our source. And for that, we have built a device that is with the electrospray capillary clamped in a copper block. We can control the temperature just with a bath, like we would do in a melting experiment. And here it's an example with the tris-Q ligand, so it's a flat aromatic molecule. This is the mass spectrum you get at room temperature. You have the quadruplex for with 2 potassium, so you can start reading this by yourself now. You have a 1-to-1 ligand complex with mainly 2 potassium, but also a little amount of this 1 potassium that we discussed before with the telomeric sequences. So these are the 3 states that we can distinguish at room temperature. Now, what happens when you heat? That's what you would do if you perform a melting experiment. And here the surprise was that when you melt the complex, it's not just that the quadruplex is unfolding. Yes, we see that. But the complex is not just disrupting as a function of temperature. At intermediate temperatures, you have very weird things happening, and especially you have the appearance of a 2-to-1 complex, although you started from the 1-to-1 complex. So at these temperatures, actually based on number of ligands, number of potassiums, we can distinguish until 8 states coexisting throughout this melting experiment. And this looked a bit strange at the beginning because it sounds totally counterintuitive. You melt a 1-to-1 complex and you form a 2-to-1 complex. But then we reasoned that at these temperatures, it's already above the melting temperature of the quadruplex. So we have here the quadruplex unraveled. So it means we started with equimolar amounts of ligand to quadruplex, but when we melt the quadruplex, the excess of ligand is becoming huge. And that is why, compared to the remaining quadruplex we can form these 2-to-1 complexes. And of course, at super high temperature, then we melt the whole thing. So then it means that when we quantify the MS signals as a function of temperature, you can have a whole range of melting curves, but they represent the concentration of each of the states that we can resolve in mass as a function of the temperature. And you can see that you have something much more complex than the two-state process that you can typically fit with a melting experiment. By difference, we can deduce the amount of ligand free because we know the amounts of ligand engaged in a complex. And now it means we can do a Van 't Hoff analysis with any of the equilibria that is involved in this complex series of states. And now when you think of it, thermodynamics is about state functions. So you need to really define the states in order to do meaningful thermodynamics. And now with all the states we could distinguish with mass spectrometry, we could disentangle the enthalpy and entropy contributions to each of the states we have. So yeah, coming back to the TRISQ observations, and actually it's an observation that we did for many ligands. Actually what we found is that the ligand binding, so here we start with this state, we set it as a reference, is the quadruplex folded with 2 potassium but with no ligands. And then we can compared to this state, that is favorable to bind 1 ligand and 2 ligand. This is delta G. But actually what is favorable in that is the ligand binding is entropically favored, not enthalpically favored. And that is something we found for many of the ligands that we have investigated in that study in 2018. So also I encourage you, although the paper is quite maybe a bit long and complex, I wanted to also highlight a couple of points in the discussion section of the paper, because I think it has important implications beyond the mass spectrometry, but for any interpretation of melting experiments. So here, actually, at the same time, we have temperature dependent, but we measure the equilibria directly for each of the temperatures. And it allows to reconcile some observations that could be, let's say, discrepancies between a melting assay and an isothermal assay. But also a consideration is that when you model what would be the melting temperature for a same equilibrium, for example, here we have, we take a quadruplex binding to a ligand, single binding site, 3 different cases, same KD value at 298. So it means same fraction of ligand bound at a temperature of 298 Kelvin. So here this ratio is the same. But what happens in terms of melting if you have different enthalpic or entropic contributions to the binding? So here it's an example of enthalpically favored or entropically favored complex. And here, depending on whether the ligand binding is enthalpically favored or entropically favored, see how the modeling of the melting temperature is going to change. When your ligand binding is entropically favored, it means it's going to be favored at higher temperatures than if it's enthalpically favored. So it means that if you have 3 ligands of same affinity, different enthalpy or entropy preference effects, you're going to have different melting temperature. And this is the reason why the melting temperature alone is not a good indicator of binding affinity at a low temperature. Another implication is, if you think about it, many ligands were selected in first screen by a melting assay. So it means, in my understanding, there could also have been a bias in ligand screening, a bias of preference for ligands that are for which the binding is entropically favored, just because it's a melting assay that has been used as a proxy for binding affinity. So it's just a proposal here, but maybe we should also revisit some of the screenings and really also focus on, let's say, direct assays, also to have a chance to find ligands that are binding in an enthalpically favored manner. Another point of discussion is that, well, the thermodynamic profiling of folding or of binding can also be useful when you don't have access to high resolution structures, because a similar thermodynamic profile for binding or folding can also indicate folding into similar families. So in interest of time, I will not go into the details of this part, but you can see the discussion in this paper. So to wrap up, yeah, I think I'm going to end here, unless there are questions about eye mobility, then I can show the extra slides. To wrap up, really take-home messages I wanted you to get away with today is that actually, because in the gas phase, ionic bonding and hydrogen bonding are strongly reinforced, and it's really the type of physical forces, the type of bonds that are important for quadruplex folding and also for ligand binding. Actually, mass spectrometry is a very favorable case for application in nucleic acid biophysics. And through many collaborations and also people who have imported the techniques in their lab, it has proven repetitively useful and reliable. The main message is that you have to think of a mass spectrometry assay when you really have a question or a hypothesis to test regarding the stoichiometry of what you're studying. And also in our experience, most often than not, we have found unexpected stoichiometries when we did a mass spectrometry assay. So I really encourage you to try it out, to convince your facility, you convince collaborators to try it out when you have complexes where you really want to get to know more about the structure and also how you can, what stoichiometries you need to fit your other biophysical assays. This is very important. So based on the masses, we can distinguish coexisting states, which is super important for thermodynamics because, as I said, the basic of thermodynamics is about state functions. You need to define your states to have meaningful thermodynamics. Mass spectrometry can be quantitative. You can determine KD values with mass spectrometry. You can do kinetics experiments. You can do also exchange experiments, competition experiments. Also with, if you can control the temperature of your source, you can do either isothermal or melting experiments. And we have shown how doing this together can help, thanks to the direct assay of mass spectrometry as a function of temperature, to help to, let's say, solve some apparent discrepancies between different types of assays. So if you're interested, if you have questions, I have a few slides of some more advanced topic, which is the characterization of the structure of each state with mass spectrometry, but it's more like an advanced mass spectrometry topic. So I will skip this for the moment and take the time to acknowledge my team. So it's a pretty recent picture of the team, but I really want to thank Adrien Marchand, who did a lot of work that I presented for you today. And Frédéric, that was not on the group picture that day, but who's doing a lot of the instrumental work and advanced mass spectrometry work in the lab. Thank these agencies for funding. A couple of references for mass spec of nucleic acids. On the one hand side, a comprehensive review that appeared in Chemical Review last year. Everything you want to know about mass spectrometry of nucleic acid complexes. There are lots of G-quadruplexes in there, of course. And a more personal perspective, then it's much shorter, published in Accounts of Chemical Research. And with that, I will be ready to take questions.